hope everybody's got a good seat. Well, I'm going to talk today about what I think is the single most incredible achievement in human history. It's an achievement that's beyond the ability of any single person on the planet to fully understand. It's an unfinished and continually evolving work, and we as humans probably started on this journey 200,000 years ago, and I would suggest we will never fully complete it. I believe it dwarfs the major inventions and structures that we normally consider to be the great achievements of civilization, with all due respect to this wonderful location we're in at this very moment. So I'm going to begin by telling you two stories. In the beginning there was darkness. Papa, the Earth Mother, and Rangi, the Sky Father, were bound together, their bodies in a tight embrace. Between them lay their children, cramped in the darkness. The children could not move and writhed and struggled to make more space, but to no avail. The children got together to discuss what to do. "Let's kill them," said some. But Tane, god of the forest, had a better idea. He lay on his back and pushed Rangi, the Sky Father, with his mighty legs until the pair parted. And slowly but surely the earth was flooded with light, bringing life to the earth. This beautiful story of creation from the Maori of New Zealand, and many others like it from all over the world, had important significance within a tribe or a group. And of course other tribes and groups had their own stories, each with their own significance. Nowadays most of us understand that these stories are simply stories, and all over the world people have told these stories in a bid to try and understand and make sense of the world around them, and to answer a question that I wager everyone here has thought of at some point in their lives. How did we get here? So here's the second story. Around 15 billion years ago, the universe exploded into existence, perhaps by way of a massive Big Bang. The universe expanded, clouds of gas condensed into stars, and planets formed around some of these stars. Our own star is about 5 billion years old, and the Earth on which we are on now is around 4.6 billion years old. Initially, the surface of the planet was hot. It was bombarded by debris from space so large that the explosions vaporized the fledgling oceans. After a time, conditions eased, and chemical reactions gave rise to simple molecules. Among these molecules were molecules able to replicate themselves, to copy themselves, and these subsequently became encapsulated in membranes, becoming the first living cells. These cells were able to harness energy from their environment, and from these evolved more complex multicellular life forms. Plants and animals emerged and conquered the land. Around 250 million years ago, dinosaurs evolved and dominated land and sea. Then, 65 million years ago, a massive meteorite hit the Yucatan Peninsula, wiped out the dinosaurs, and a new group came to prominence, the mammals. The mammals diversified, and some of the members of one group, the primates, learned to walk upright, built tools, harnessed fire, and were able to talk and communicate. They originated in Africa, and subsequently they spread across the globe, invented art, told creation myths, wrote these down, and later on invented science, technologies, traveled to the moon, and finally became sessile, using only their fingers and their eyes in order to operate their mobile devices. This is an utterly brief account of the history of life on Earth, and excepting a bit about the smartphones, it's pretty much true to a children's book my son has. If I were to present this account at a scientific meeting on the origin of life, I can guarantee that absolutely everyone in the audience would find something to disagree with in that account. Partly because I let slip that it came from a children's book, but mostly because the details of all the ideas espoused in that account are subject to ongoing debate. In keeping with the Brain Without Walls theme...
Slides are coming.
Yeah, I know that's okay, it's deliberate. In keeping with the Brain Without Walls theme, I want to give you some insight into this process, by way of some questions my collaborators and I have been addressing. While the emerging view of our origins is fascinating, I believe the process we've developed to help shape that view is crucial, and I hope it will give you a take-home message that's of relevance to your own interests. So as I'm sure you're all familiar and shown in this slide, genetic information is stored in the form of genes in DNA. In order for that information to be used, it needs to be expressed. and that happens in a two-step process as follows. The first step is to make a working copy of that information, something that we call a message, and that's made of a slightly different molecule called RNA. The second step is that information is translated, and the information is read off the message. That message carries instructions that enable the production of a molecule that does work. That type of molecule is called a protein. So proteins do all the reactions in our bodies or in any type of cell. This process is something that we find in all life forms, everything from the trees around us, every person here, even bacteria that sometimes we pick up infections and so on. All of these life forms do the same fundamental process. Another interesting point here is that DNA is effectively it's just a storage medium, and it has an interdependency with proteins. So the information for making proteins is stored in the DNA, but in order to maintain and copy that DNA, proteins are required. So we have this classic problem that scientists love to hate, the chicken and egg dilemma. Which came first, the DNA or the proteins? Given each is required to make the other, how do you progress? Well, how did that situation arise, and from what did it emerge? A hypothesis was, considered in the 1960s, which is as follows. Once upon a time, RNA, remember this message that I talked about, performed both of those roles. It was both a genetic material and was able to do work in cells. So this was a hypothesis, and it would implicate, would mean that early cells would operate as follows. This is the same process we just saw, and instead of having DNA and proteins, we have RNA as a genetic material, working copies of RNA, making RNA molecules that do the work, and are required to then copy that RNA. Do we have any evidence for this RNA world? In terms of the genetic information, yes, probably some of you have come face to face with that in the course of, I can hear some coughs, so that may indicate that, in the course of this year. If you've been infected by influenza, hopefully no one's been infected by HIV, or if you've been unfortunate enough to know anybody who's had the SARS virus, these are all RNA viruses. That means their genetic information is carried on RNA, not DNA. We also know that RNA can operate as enzymes. In fact, 2 Nobel Prizes were awarded for this discovery during the 80s. And in fact, if we look very closely at this process that I explained to you about gene expression, it turns out that the modern machinery for making proteins has at its core RNA. Okay, so here's a schematic of that, and we have 2 functions that are going on in this picture. So this is, at the bottom here, a way of reading the information in the message. This part of the reaction is done by RNA, and the other part here, the reaction of making proteins out of the building blocks, is also performed by RNA. So at the core of this machinery is RNA, which is required to make proteins. Okay, so RNA is making the proteins. So there are no proteins involved in these reactions. Okay, so we have an interesting situation here. This is exactly what we would expect from this RNA world model. If you're going to move from RNA to proteins, you'd expect an RNA machine to have been invented in order to invent proteins, if you like. But there's another problem, and that's this. How did the messages originate? How do we go from no information to information? This is what a modern gene looks like inside your body. We have, I'll show you what these parts are in a minute. So remember, we have the DNA that has to be transcribed to make a message, but an additional process goes on in genes that are expressed in your body or in the cells of a plant, and that's something called splicing. So anybody who remembers cassette tapes will know what this term means. What happens is that these blue blocks here, those carry the information required to make the protein. So those pieces are spliced together and can be read, and you can make a protein. The intervening pieces that are in dark blue here are actually, they're excised, and then what happens is that they get degraded. Okay, so we have sort of some superfluous information in those intervening sequences. Another important fact here is that splicing reaction is carried out by an RNA machine as well. So that is another case of RNA doing the work in the cell. So that possibly that could be as old as the machinery that's doing the last step here, the translation process. protein synthesizing process. More recently, we've discovered that there are genes that have two products. So this is the same picture we saw before, but sometimes in those intervening regions we find extra information. And what happens with that extra information is shown as follows. We get our working copy made again, but with this time we get two products. The first is our message that we can translate into protein. The second thing is that those black boxes are actually information for producing RNAs that go off and do work in the cell. So we get two products for the price of one gene, if you like. If we think about RNA preceding DNA, we might ask, if we flip that whole picture of the way we think about genes around, that they're there for making proteins, and we know that there are RNAs hiding inside these intervening spaces, could it have been the case that the genes were inside out early on in the evolution of life? So how would that look? Would it look like this? Here's our precursor. We get our RNAs that are excised from those intervening regions. Our message, however, instead of being translated, is just discarded. Okay. Do we have any evidence that that's a viable process for genes? It turns out there is good evidence for that. So this is a gene that is in every one of you. This is a gene from humans, and it's a very complex looking gene. It carries 9 different small RNAs, which are in these colors here, and the gray pieces that are spliced together, which would normally make a message. So there's our 9 RNAs, and there's our message. What happens here, though, is that that's just simply degraded. So it's not used at all by your cells. It's sort of superfluous material. So this is a case of a genuine inside-out gene, exactly as we might expect from an early system before proteins. What we know from our studies of genomes is that this kind of material is being expressed, and it may be superfluous. There's lots of material that sort of gets produced and then discarded. But it can also get picked up and used, just like a tinkerer in a workshop trying to produce something in his spare time may find something sitting in a corner and make use of it. So too, this happens in evolution. And we find that those superfluous pieces can be recruited into a new function. So an example of that is our immune system. We wouldn't have antibodies if we hadn't picked up a spare piece of material that was lying around doing nothing. In fact, it was doing something a little bit malicious. That's another story. But that was the reason why we have that capacity at all, is because we picked up some genes that weren't at all involved in that. So this process of tinkering and recruitment can generate new genetic information. And this is happening all the time. A key thing in order for this, so what I can say at this point is that all of these processes seem to make sense. They fit well with the idea of a very early evolution of a genetic system based on RNA alone. But in order for that to, carry any weight, it also needs to have a historical trace. We have to be able to say that this is a very old arrangement. So how old is it? One of the ways we can look at that is to say, if we look at the oldest genes that we know of, the genes that trace right back to the common ancestor of any cellular life form on the planet, so we're talking about the organisms that live in extreme environments, at the bottom of the sea, or plants or animals, genes that are present in absolutely everything, do they show this arrangement? It turns out that we can do that by comparing genomes. We can say, let's take some human genes that have this arrangement of 2 products in one gene. Do we see this elsewhere? We can look through thousands of genes and find that, yes, if we look at humans and mouse, there are hundreds, maybe over 1,000 genes that look like this. If we go as far away as chicken, we see exactly that same result. We may find that it's maybe 200 or 300 genes. However, if we go even further back to, say, plants, we can say the arrangement still exists. We still find this pattern. However, the colors here are representing a slight change in the information, and that is that these little RNAs that are sitting in there, these genes inside the genes, are actually different RNAs. They're unrelated. So that tells us 2 things. One is that the arrangement is old, but there's a catch. It turns out, I can't show you the details, but it turns out the RNAs are actually capable of moving around. So one of the things we know about our genome is it has a lot of the genes will jump around. They don't stay in place. They migrate from one chromosome to another, and this also applies to these RNAs. They will jump from one gene to another, which means that this, from the point of view of showing how old this is, the information may well be lost because of another process that's happening. So there's this process of migration, if you like, resulting in erasure of an earlier signal. So if that signal was there, we can't detect it. That's a pain because it means we can't really fully test this hypothesis, and it could actually be completely wrong because we have no direct evidence for it. Everything we can see supports it, but it might not be wrong. It might not be right. And actually, my PhD student who has been working on this and took great glee in slaying this hypothesis, to which I responded with this quote from T.H. Huxley, which is the great tragedy of science, in his opinion, was the slaying of a beautiful hypothesis by an ugly fact. And you'd think that this is a time for someone like myself to be very dejected, but that's to miss the point of science, and I think this is the idea that I want to get across to you. If we think about it, each slain hypothesis or idea paves the path to new ideas and discoveries. So more than any other thing, this understanding of where we came from and how we got here, one, it stands as a testimony to our civilization. It's a feat that required huge leaps of technology, advanced chemistry, telescopes, sequencing genomes. It's also a global question. It's a question that everybody asks in every culture and every civilization, and at some point every individual will want to know this question, how did I get here? Which is why my son has a picture book about this and finds it fascinating. Minds from all over the world are collaborating to help improve our answer. So my picture book answer is almost certainly not completely correct, but we're changing this globally as a species, because there are researchers all over the world trying to change this, and every day hypotheses are slain. This is not a tragedy. This, I think, is an incredible societal brain without boundaries. So if there's one take-home message that I think you can all learn from this, or we can all learn from this, it's a good thing to be wrong sometimes and to admit it and to say, what does that tell us and where can we move forward from that? Thank you.